I don't think I need to introduce Terry. He basically founded the fields of first computational neuroscience and neuroAI, and then what other field he's going to introduce today, we don't know, but it's going to be fascinating. So Terry, please.
Wonderful. So I was once introduced at a big distinguished lecture by saying, Terry Sejnowski needs no introduction. So let me tell you a little bit about myself. Well, I've been here in San Diego for about 35 years, and this has been an extraordinary place to do science. And especially, you've heard over the last few hours a series of talks that I could not have asked for a better background for the story I'm about to tell you. And it's about a part of psychology that hasn't really been cracked yet at the neuroscience level, at the level of mechanism. It's called long-term working memory. Now, if you look up working memory in the neuroscience literature, typically they talk about remembering something over a few seconds, but that's not what psychologists mean by working memory. So here is the kind of a very simplified version of the different kinds of memory that have been observed psychologically. This is behaviorally. Well, first of all, there's a very fast sensory memory, like a trace, and it only lasts very briefly. We're talking about less than a second. But that can go into a short-term memory that can hold on to it for longer. But now, this can go too. The short-term memory will decay unless you put it into this box called working memory. And this working memory is what is happening in your brain right now. I'm going to be talking for about 35 minutes, and you're going to be hearing one word after the next. You're going to be seeing one slide after the next. And at the end, you're going to be able to ask a question, which means that you've remembered what I said, and you remembered maybe one of the slides. Where in your brain is that stored? How do you access it? How do you manipulate it? And this is a very simplified version. You have a phonological loop, and by loop I mean that it's repeated or somehow is refreshed. Some of it by rehearsal, and then there's a visuospatial, which is images that you can remember, and then episodic buffer. And these are all psychological terms, right? Central executive people usually say that's somewhere in the prefrontal cortex, but how they interact with each other so that you can actually think is not something we understand at the mechanistic level. Now, some of this information gets encoded into long-term memory. We had a great discussion earlier in the morning about how that might be done perhaps by these ripples. That would be one of the candidates. But I want to focus here on what's going on in the working memory. Is there a possible way to understand this in terms of things we're familiar with? So first of all, if you want to really think about the kinds of networks that are probably likely to be able to hold on to a lot of information over time, recurrent neural networks come to mind. And in fact, recurrent neural networks were very popular about 3 years ago for doing language translation. So here's how these recurrent networks were designed. Okay, the idea is that this is all one network at different time steps. And the words come in one at a time, just like you're hearing me, right? But it could come in from a book or text. They're embedded into a vector, and then they circulate. And so the idea is that the word at the beginning might interact with a word at the end, especially if you're doing German, in which case the verb always comes at the end. So you have to have a trace of the first present at the end. And then there's a cue, and then you've trained it to be able to come out with a sequence of words in another language. And this actually works extremely well, by the way. I mean, it really was state of the art just like 3 years ago. But now here's the problem. There's an exponential decay. When you put something in here, it's going to decay as you go forward in time. And so it really is, you're limited here in terms of the length of the sentence or to go beyond that, right? Well, we now, we can now think about how does a transformer do this, right? Transformer can take in huge sequences and be able to take them in and somehow manipulate the internally and be able to, at the end, not just take in all of the words that you've just given it, but also output a lot of words. I mean, that's really, I think that's remarkable. So, well, first of all of these inputs here, whether it's a sequence of words or a sequence of the spoken words or text or videos, they're all converted into what are called vectors, vector embeddings. And it's just a long vector that is associated with each word or token of a word. And that's going to be the input to the transformer. So here's how a transformer translates from one language to the next. So you have words coming in, just like you did before, and you embed them, actually just like you did before. The difference is that they're not coming in sequentially. They're all concatenated into one long input vector. And now that whole vector goes into the encoder. There's a little magic box here, which is self-attention. I'm not going to talk about that, but it associates different words across the vector. And then it goes into a decoder, which then sequences chooses a word, and that then is added to the long input vector. It gets longer and longer and longer. It's trained, by the way, simply to take in text, long sentences, paragraphs, and to predict the next word. That's all it does, predict the next word, predict the next word. But once you do that, now you just turn it loose, and it can take the input as a query, for example, and produce one word after the next in response to your question. Okay, so there are two ways you can hold on to information over many minutes and hours, right? Long-term working memory works over a time course of hours. One is you have some kind of circulating electrical activity that actually is able to hold on to the information. Is there potentially a connectivity pattern in the cortex which helps hold on to information over time? The other possibility is through biochemical reactions in neurons, which are slow and can last a long time. For example, short-term synaptic plasticity. Maybe you could hold on to information even longer. So let's start with trying to improve on the recurrent networks. So this is work that I did with Max Welling and his then student, Andy Keller. And the idea is that we put some inductive bias into the network. We don't start with a network that's small random weights, which is traditionally done in machine learning. What we're going to do is have a network, a connectivity network that looks like this. There's going to be ones along just above the diagonal and zeros everywhere else. You might think, well, why do that? Well, the reason is this is a discrete version of this partial differential equation for waves. This will produce waves moving in one direction. And here's the actual network. It's a traditional network in which you have a variable h, which is goes through this process of integrating across the input vector here, and then it just cycles right over time, one step after the next. So, first of all, you get much more coherent output from it's called a convolutional RNN because it turns out that matrix is equivalent to a temporal convolution. But you can see this is time, and this is one-dimensional array of units. You can see that it produces very nice traveling waves, and they're random because nothing has been trained yet, as opposed to the initial condition of a randomly initialized network, which it doesn't have any structure. So what can we do with it? Let's do a simple task. We're going to try to copy an input pattern that comes in. It's a binary pattern, like 10 different inputs, like playing a piano. And then at this point here, it's got to repeat it on the output. And it gets it right. Now, this is for the wave recurrent network. What about for the traditional randomized neural network? Well, here it is. Here comes the input. The output already is beginning to be a little ratty here. It's not quite the same. And now we can make it more difficult. Let's have a time delay, 10 steps. It does it perfectly. 30 steps, does it perfectly. In fact, you can do it for 100 steps. You can do it for 200 steps. It will just circulate, 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 and it can hold on to information, sequences of information for enormous lengths of time. And not only that, and by the way, the random network is just completely hopeless. Not only that, Here's the learning curve. This is the mean square error on a log scale. Well, here's the wave network, and you can see it goes down immediately 2 orders of magnitude, and it keeps going down here, 8 orders of magnitude. It's getting it perfect. Whereas, these networks that are randomly initialized don't really get off the ground. They just are very, very slow to train, and they never really get the task. Now, not only can we reproduce a sequence, replay, we can also ask it to do things like, for example, add numbers together. So we're going to give it a sequence of numbers, 2, 22, 3, 1, 5, and we're going to ask the network occasionally, because it's getting a lot of input, to simply take the queued numbers and add them up, right? So at the end, you give the sum, and you train it to do that. And here we go. It turns out that with the wave network, here's the test error for sequences of input that goes up to 1,000, and it just does it perfectly. It's really amazing. Whereas, as you can see, for small sequences, the randomized network actually eventually gets it for small sequences, but for long sequences, it's hopeless. So this is really an important insight, because it means that not all recurrent networks are created equal. Some networks are better at some tasks than others. And you've already seen this picture. Can anybody remember where it came from? I know there's one person in the room who can recognize it. Two. Okay, I'm the third. Now, this was for one of Mark's papers, and I have to say that, Mark, do I have permission to show this? Okay, I just want to make sure, because I'm not sure if it's published. Is it published?
It's on the archive.
Oh, okay, it's on archive. Okay. But actually, it answers one of the questions I asked Mark, which is, here's a recording from one location, and you can see that this is a gamma, so it's about 40 hertz. And you can see at the 3 locations, there's a phase shift, but you can see the whole thing only lasts for about 100 milliseconds. So it's a packet, at least for gamma, at least in this case. This was in V1 mouse. Now, I said before about inductive bias. Well, what's the inductive bias in the cortex? It turns out that in the cortex, if you look at the connectivity within a column, that locally it's excitatory between pyramidal cells, but then if you look at the next column, it is suppressive. And it's not through an inhibitory synapse, it's through the inhibitory neurons in the next column. It has the shape of a Mexican hat. If you lay out the connections in a diagram like this, pre versus post, it has this diagonal form, which you will recognize, very similar to the diagonal form that we saw in the previous case. And the difference is that it's symmetric, right? The other one was not symmetric. And that means that this one can support traveling waves in both directions. And it's a second order wave equation, not a first order. So it turns out that the inductive bias during development in the cortex is for waves, and that's what we see. We see them during development, we see them in adults, we see them changing, and they're very rich. By the way, they're still called oscillations by most neuroscientists. They're not. Every oscillation is a traveling wave. Then Yuri, you can back me up, right? And Yuri is Mr. Oscillation. He has studied oscillations deeper and more thoroughly than any other neuroscientist. And I have to say, I've inspired by the research you've done that has led to this, this approach to thinking about what is the cortex really good at. And by the way, this connectivity also gives you a line attractor. It's not a point. No matter where you start, you're going to end up at this trough, and a little push will push you along because it's a neutral equilibrium at the bottom, and that's why you get a traveling wave. Well, we modeled it with a ring attractor, a traveling wave in a ring of connected neurons with a Mexican hat connectivity, and we could not get, we can get waves out. It's very easy to get waves out that are dense, where all the neurons are active in a little group as it goes around. But this is very difficult in the cortex. It's sparse waves. In other words, only a small fraction of all the neurons, as wave front goes through, are actually spiking. So it's sparse. And it wasn't, so we said, maybe it's because we have the network's too small. It's only a few hundreds, a few thousand. So we kept getting it bigger and bigger, and this is Lyle Muller started realizing he has to microcode it, and eventually got to the point where we had 100,000 neurons. And then something magic happened, and it was absolutely astonishing. I just love watching this. It's so peaceful. This is the membrane potential, and this is a spiking, or integrating spiking model of neurons. And you can see the beautiful waves, and because they are sparse, they go right through each other. So this is really much more closer to the actual scale of the cortex. 100,000 neurons is still small, but it tells us that there are things that happen at scale that don't happen when you have small networks. And 99.9% of all the models that I've worked on, and almost everybody else in computational neuroscience, have been relatively small networks with a few hundred units, and you can't get sparse traveling waves out of that. Okay, and as you can see, the power spectrum and the spectral analysis and spatial temporal frequencies, that's the velocity of the traveling wave, that slope. Okay, so what good is a traveling wave? And this is something that I've been thinking about for a long time. I have a paper in Nature Reviews of Neuroscience in 2018 with Lyle, and we listed a bunch of possible ways that traveling waves could be used, and John Reynolds is here. Where is John when you need him? No, we had a paper in Nature showing that, in fact, spontaneous traveling waves can affect your perception of a weak stimulus at threshold. This is now in a monkey and looking at MT, traveling waves through MT. At the right phase, you can see it, and at the wrong phase, you can't. Well, here's another potential function that we brought up briefly, which has to do with recoding the input. So here's a visual input. It's a spot of light, and it goes off. But we know that a traveling wave this can generate a traveling wave that will then expand over time. And you might think that you've lost all the information, right? You know, the cells out here aren't telling you very much about this. They're looking at it in the past, right? It happened a long time ago. However, if you have a population of neurons, by looking at the center, you can exactly show where the center is. So that means the information is there. It's just been recoded. It's been recoded into a wave. Not only that, but you can even know when it occurred because of the time delay. If you look at the radius, that tells you how, and you know the velocity, you know when it occurred. So everything about this stimulus now is contained in this population. It's a population code that is different from the traditional ones that neuroscientists think about because it encodes both space and time. Spatial array of neurons that have information about the spatial and temporal input. And if you have more than one input, as you can see, you get interference fringes. And it gets really hairy and complicated, and it's not intuitive. You look at that and say, I don't know what that is. It's like a hologram. You look at a hologram, and you don't see anything until you put a laser on it, and suddenly you can see the object. And it was coded in terms of the interference fringes. And this is, we call it a space-time code, right? You have input coming in to A, both directly. That's the strong thalamic input. You also have input coming in from neighboring neurons, but at time delays. So A is mixing what's happening right now with what's happened recently in the past at a neighboring location, and in fact, all the neighboring locations are going to be contributing. So this is a really complicated population code. I mean, it's very complicated. And just because we can't decode it doesn't mean that the brain can't, right? So it's there. The information is there. Not only that, but if you now go from V1 to V2 to V3 to V4, we know that the receptor fields get longer, and now you're passing up this population code, which represents time intervals, and that now gets longer and longer and longer, and by the time you get to the prefrontal cortex, we know from fMRI that now you're dealing with time scales that are on the order of a minute or two. But that's not the same as an hour. I don't think I'm not sure. I think this population code can hold on to intervals, up to, maybe a minute or maybe longer. But at some point, you need to have another mechanism that takes you from a minute to an hour. So let's just, here's some predictions. Space-time codes are dynamic, not static like short-term memory. Space-time codes can be tested by decoding time intervals. People decode inputs. They give a flash input, and then they try to look at the activity patterns and then predict what the input was, right? Jack Gallant has done a lot of that. Well, why not give it a movie and see if you can reconstruct the past? What was there before? This can be done today if you just had the data. The time window expands up the hierarchy. And we know that the complexity of the patterns that neurons respond to become more complex, and so why not the space-time codes? Now, you've heard about the hippocampus. And people have recorded from it. There are Nobel Prizes have been given for the place fields. It's a glorious story about this little structure at the very top of the pyramid. All the sensory inputs, are focused into it, so it really has the highest level of representations. But Thanosiopis, and this is now, we're talking about, let's see, like 15 years ago. Notice when he put an array of electrodes in that it wasn't synchronous across the whole cortex. So these are electrodes and the false colored so that you can see the peaks, for example, flowing from the anterior to the posterior. So there's about a half cycle across this. This was confirmed by Urey, was it?
No, because they said it's 360 and we showed it's only 180.
I was about to say that.
Big difference.
No.
The entire theory is wrong.
No, I was going to say confirmed the traveling wave, but they made an error, and Urey was the one who fixed it. Okay, so yeah, it was that I was literally about to say that. Okay, so here's a very, very brief interlude about what might be going on in the hippocampus. It's a whole paper in Neuron published a couple months ago. Here's the basic circuit. The input from the upper layers of the internal cortex go into dentate, from there to CA3, which is a highly recurrent network, from there to CA1, the output. Now, CA1 gets two inputs. It gets an input from CA3, but it also gets a direct input from internal cortex. And so here's our hypothesis. Our hypothesis is that CA3 has to remember sequences coming in, and one way that you can train a network to remember a sequence is to be able to predict what the next input is and compare it with the actual input that is coming in next. And from that error, you correct the weights and therefore get better and better at remembering the sequences of inputs coming in, right? And then you can replay it. You can now give it of the first input, and CA3 should be able to replay the rest. And we know that happens, so this is a way of actually achieving that. So here's the model. It's called a predictive autoencoder. An autoencoder is one that takes some inputs, puts it through a bottleneck, and tries to reproduce the very same input. Well, here we're trying to not reproduce the input, but the next input. So we have a different loss function. I won't go into details. We also replaced the hidden units here with a recurrent neural network. And this is work that was done by UC Chen when she was a postdoc, a graduate student. She's now a postdoc at University of Washington. So here's an example. We have 9 movies. They're called sprites. Each figurine is different, and they're doing a different action. We trained it up on these 9 different figurines, and then looked at the input, looked at the activity patterns. Everyone says that these networks are black boxes. Not true. You can look in. I mean, there's nothing preventing you. And if you look in, you analyze them with independent component analysis, what you show is that each one of these movies is really part of the space of activity patterns, separated out so they don't interfere with each other. This is a tiny network of 200 rate-coded neurons. So this is really, it's saying that, you can, with the right kind of a learning algorithm, you can actually store a lot of information. 9 different movies. Now, here's the other shocker. Not only can we look at the activity, we can look at the weights. Now, what do you think the weight matrix looks like? Well, here's for 2 different stored inputs. They're both sparse. These are the weights, presynaptic, postsynaptic. Mexican hats. We didn't put that in. It figured out that in order to store a lot of different sequential patterns of inputs, you use a Mexican hat, traveling waves.
What were the initial conditions?
What?
What were the initial conditions for the weights? Random?
These were randomized. Yeah, we were back in the dark ages when we started the project. Right, we didn't initialize it with the waves, but if we did, this would have learned a lot faster. But it discovered, actually we learned something, it discovered the wave solution, right? It does beautiful. You give it the beginning of the movie and you see the end. Now, here's something that Dan Feldman once mentioned to me. This is when he was at UCSD, now at Berkeley, is that, Terry, the neocortex has these layers, and the function of layers and the connectivity seems to look a lot like the hippocampus, which has discrete regions. And L4, layer 4, which has a direct input from the thalamus, sparse activity, just like the dentate, that gets up to layer 2, 3, which is very highly recurrent. And then from there, it goes down to layer 5, where it also gets an input from the thalamus. And people always forget this, but it turns out that it looks just like the convergence of a predicted input. And so it could well be the story I've just told you is actually going on in each layer of the cortex, in the upper layers 2, 3, and it's stacked in a hierarchy. So really what the cortex might be doing is not just taking snapshots, but it's taking in whole sequences and representing them, not just static inputs. Okay, and similarly, it could be that the cortex is just a stack of predictive recurrent autoencoders. I mean, this is just hypothesis, but if it's right, it really gives us a different picture. Time is an essential part of this picture. So more predictions. Waves in the cortex encode temporal context. A temporal context is really important. It's what I was talking about earlier about connecting the first word with the last word, to understand what the words mean. Waves in the hippocampus are generated by Mexican hat. Nobody's measured that yet. This is a real prediction. I don't think anybody would have guessed this. Yuri, would you guess this if someone told you? It doesn't look like it. It looks like it's randomly connected, right? It's associative. It's called archicortex. It's called this kind of an associative network. But this suggests that it's actually very highly organized in order to get the waves, right?
The same day bone neurons are connected better than different day bone neurons. So there is a
you're talking
skeleton.
about in the dentite.
In CA3, CA1, everywhere.
Oh, you're talking about during development.
Yeah.
Oh, okay. Okay. Well, maybe that's how it's put together. Okay. Okay. So let's get back to long-term working memory. So how can we extend the time from minutes to hours? And what other neural mechanisms? And this is really, I want to make a suggestion, a plausible suggestion, and I'm going to use as the mechanism spike time-dependent plasticity. It's been very well established in slices. This is from the Discovery paper, 1997. Slice recordings from two neurons that had connections between them, shown in little blue and kind of yellowish dots here. And here's what you do. You pair the presynaptic input to one neuron with a postsynaptic firing, like here. You have the, here's what I want to show you first. Here's the EPSP coming in just before the delayed postsynaptic action potential that goes up the dendrite. And you pair that at frequencies that range from, as you see, 10 to 40 hertz. And you have to pair it, like typically 5 bursts of 5 spikes at different frequencies, and you do that 10 times, spaced by 4 seconds. And lo and behold, you have an elevated potential that lasts for at least here about a half an hour. Now, there's something really interesting here, which I didn't pick up when I first read the paper. If you look down here, if you do the pairing at 2 hertz or 5 hertz, nothing happens. You have to be at least up to 10 hertz or above 40 hertz. Does that remind you of anything, that frequency band? That's the frequency band for traveling waves that we all know and love, right? Could it be that the purpose of the traveling wave is to trigger STDP? Now, what's interesting about STDP, this elevation is always thought to be a mechanism for long-term memory that will last for days and weeks. And if you really buzz it, it might, but maybe not in the cortex. In fact, what if you just give it one of the 4, one of the 10 pairings, a burst of 5 at 40 hertz? Well, it turns out that with one pairing, one burst, it lasts for about 20 seconds. It goes up very quickly, lasts for about 20 minutes. If you do it with 2 or 3, it may last for an hour. If you do it with 4, now we know it can be more than an hour. But it is temporary. It's not permanent. But no one has shown that it's permanent. But it's in the right time scale for long-term working memory. And in fact, the mechanism you need for long-term working memory, You don't want it to be permanent because you want to erase it. You want to reuse it because you're going to be, the next day you may only remember a few things from the lecture that you may have attended, but you don't want to remember every detail, otherwise you're going to go crazy, and there are some people who actually can. So it shows you that it's not functional to do that. But it might be helpful to actually look at their traveling ways. Okay, so the beauty is if you do the pairing with different time delays, you can get potentiation if it's in a time window of plus 10 milliseconds pre, before, post, or it could be minus 10 milliseconds post, before, pre. So you can move it up and down, and so you could use this to be able to imprint a lot of synapses with what's happened recently from incoming information that have stimulated stimulus-dependent traveling ways. And this can be done at every level in the hierarchy. So here's the working hypothesis. Temporary STDP in cortex is induced by traveling waves. The stimulus-induced traveling waves create long-term working memory. Spontaneous traveling waves, interestingly, as actually Mark mentioned this, they just happen spontaneously. And maybe that's kind of a refresh, to keep the memory in the synapses, right? Because they decay too. And this is really what I think is important for attention. We know that for something you want to remember, if you don't attend it, you're going to forget it. But if you do attend it, we know that acetylcholine is activated, and we know that that produces a gamma, a state of gamma. So this could be stimulating gamma waves that then imprint the recently attended thing that you're interested in. Okay, so it's another question. Which synapses are involved in long-term working memory? So this is a study was done in vivo for the distribution of the synaptic sizes. It turns out the size of the spine head is very highly linked with the strength of the synapse. And you can see that there's a wide variety over an order of magnitude, actually 2 orders if you had a bigger sample size. Here's the distribution, but if you go to a log scale for one neuron, you can see it's log normal, and this is something that Yuri Bujaki's written a whole book about, Inside Out. It's not just spine sizes. It turns out everything is log normal, but in any case, activity patterns and so forth, and this is for a lot of neurons. So log normal, what does that mean? It means that the vast majority, like 80–90% of all the neurons are tiny, like that. And then there's only a 5 or 10% that are really big. And we know that the ones that are bigger are the most robust, are the ones that last longer. And the ones that are small, we know that they're labile. They come and go. There's new ones forming all the time and going. And that could be 80–90% of all the synapses in the cortex, all the excitatory synapses in the cortex. And if you just think about that for a minute, that's the vast majority of all the synapses. And we also know that by virtue of the fact that the biochemistry inside of the spine head, when calcium enters, it triggers either LTP or LTD, depending on the concentration. It turns out that the small spine heads, for the same amount of calcium coming in, have much higher concentration, which means that they can change their strength much more quickly with many fewer stimuli. Right? We've simulated this. Tom Bartol, is he here? Where is he when I need him? He does these M-cell simulations. So this is done with particle-based simulators that can actually keep track of all of the molecules that are important. And so we've done the simulations. Now, this is the last little piece of the puzzle. Oh, I should just to connect to what was said earlier. There are a lot of pieces here that are missing, and one of them is mentioned earlier, which is of all the things you're going to remember, which are the most important? How do you determine what's most important? Well, attention is one of them, but there may be other things that are tagging things that are important for you. You may not be aware of what they are. And this could be what a ripple does for you when you're awake. And then, of course, when you fall asleep, the ripples are still there and they can do their job, just the same way they might do it during the day, except they're doing it in a way that is going to form long-term memories. Okay, so here's one of the things that Eric brought up, which is we're looking for mechanisms that can help organize activity throughout the cortex. I meet a new person, I see their face, I hear their name, you know, maybe I shake their hand, all those sensory systems are integrated to the memory of that person. But they're distributed all over the cortex, so there's got to be some way you can tie them all together. And that's a mystery. Eric has a possibility of synchronous activity. That's a possibility. There are others, but one of the things that this has led to is the idea of what's called a global workspace. This is Bernie Bars, actually, I think, had the first generalized concept. And here's his assumption is that the sensory input comes in, and then some central core, it could be associative cortex, highly interconnected, is now going to be activated. And in fMRI experiments, this has actually been measured. You give it a stimulus near threshold, a little bit above threshold, you get visual activity in the back of the brain, in the temporal lobe, and then that spreads to the prefrontal cortex after time delay, as you can see here, you know, hundreds of milliseconds, and you get a big burst of activity here, right? So this is interesting. If it's below threshold, you don't perceive it, it doesn't make it all the way, and this is called, he calls it the, what does he call it? Ignition. It's like, you know, either you ignite a flame or you don't. And now once it ignites, though, because of feedback connections, you can have information traveling in both directions. And so in this particular scenario, what we would say is that, well, you know, the feed-forward input is creating traveling waves, at least all the way up to the prefrontal cortex from the different sensory systems, and those traveling waves are interfering with each other, and they are loading all of that information into this huge storehouse of small synapses. Now, you can use those small synapses for thinking, for cognitive operations, because it's a perfectly good network. You've created it, whole cloth for this purpose, just right now. But you don't necessarily want to hold on to that cloth, you know, for too long, just enough time to remember what happened at this talk. That's the important thing. Okay, and in this, memories are consolidated, and I brought this up. This is, again, Lyle Muller. This is our activity from electrodes implanted in the brain of someone who is prone to seizures. And as you can see now, these are particular locations. And if you look at the red dot, that is this one here. And you can see that they're all going up and down. This is a sleep spindle, again, 10 to 14 hertz. But you can see that the peaks are occurring across the cortex with phase relationships that make it look like it's a traveling wave. Okay. There it is. Okay, so follow the white dots. Okay, they're going down here to temporal lobe, they're coming up here to the occipital, to the parietal, to the prefrontal, and around it goes. Okay, so this may have something to do with memory consolidation. We know spindles are involved, and so maybe this is, again, spike time neuroplasticity, but maybe on a grander scale. Okay, more predictions, 2 tiers of processing. Fast sensory motor processing, which has been studied with rate coding, very, very well established for things that are happening over a second or two. Long-term working memory is a different tier. It's really on top of that, and it's the temporary. It's not permanent. Only parts can be permanent. But it's, you know, the hypothesis is induced with these traveling waves. And finally, this is really very testable. All of this is extremely testable, and we'll know. Okay, so this is sometimes called the global workspace, and it's thought to be the substrate of conscious awareness, according to Bernie Bars. We'll have to wait and see. Now, I want to end with something that was absolutely astonishing. We all know and love the Hebb synapse, right? You've heard about the Hebb synapse. Well, I was asked to review his book in 1999. The book was published 50 years earlier. So Neuron decided that, you know, Neuron didn't exist back then, so this is an important book. So I actually, for the first time, sat down and read it. And it was shocking, because my conception of what drove him to give this hypothesis for it's called the neurophysiological postulate, is completely different. You know, when an axon of cell A is near enough to excite cell B and repeatedly or persistently takes part in firing it, Some growth process or metabolic change takes place in one or both cells such that A's efficiency as one of the cells firing be increased. This is kind of it's viable for, neurons that fire together, wire together. I think it was Carla Schatz who first said that. Now it's in the newspapers. Of course, now there's neurophysiological evidence for it and so forth. So this is the only time in the whole book where he actually says this, right? This is like a little piece of the book. So the question is, what was driving him? And I always thought it was, this is associations. We're talking about associating stimulus with response. He hated B.F. Skinner. He hated B.F. Skinner. Thought B.F. Skinner was way off the beam in terms of trying to understand cognition. And what the problem he was trying to solve was the persistence of a sensory input. He wanted to know when an input comes in, how is it you can remember it, right? And he had this idea, which he called a phase sequence. And the idea was that it goes from neuron to neuron to neuron. And now there are 2 diagrams in his book. This is one of them. Now remember, it's 1949. It's pre-Hodgkin-Huxley. It's pre-Hebb. Pre all the modern techniques we have for looking at pathways. Tracing pathways. So this is V1, area 17. Here's an input that goes from V1 to V2, and we see that it connects to a bunch of neurons in V2, and then it also goes to neurons that go back. Feedback. He knew about feedback connections. And so his idea is, well, look here, this could circulate. This might be the source of persistence. Now, here's the other thing, and this is amazing, and this is what I wrote about. For me, it was a revelation. Let's actually see what he said. When an axon of cell A is near enough to exciting cell B, well, the only way it could possibly do that is if it actually had its input before cell B spiked, right? This is a statement about causality. A helps or causes B to fire, right? If it occurs after the postsynaptic spike, it could not have contributed. So he already had it in mind, this idea of STDP. He didn't have the negative side. He didn't have the long-term depression. But, it's clear that his thinking was much more clear than ours was. I mean, he really thought through what the problem was and how to fix it. Okay, so these are the people who have really made this project go. I've told you about the work that was done by Max and Andy on these RNNs, recurrent neural networks. We have this paper that came out, Neuron, on the hippocampus model with UC and Mia Cameron, who's a math major here at UCSD. And we have a TINS paper, which is TINS in neuroscience on this idea of a space-time code, and that was done with Lyle and Pat Churchland. And finally, I would be remiss if I didn't acknowledge my mentors. So John Hopfield was my thesis advisor back at Princeton, and my first collaborator was Jeffrey Hinton, and I couldn't have asked for a better collaborator. And just recently, they won the Nobel Prize in Physics. Now, Jeff and I worked on the Boltzmann machine. The Boltzmann machine was inspired by Hopfield's network, but Hopfield, his goal was to try to get these local memories, right, these point detractors. And we were trying to find global minima, and so what we did was we heated up the Hopfield network so that it was probabilistic, and then we gradually cooled it down, and that would give us a global minimum. Now, along the way, we discovered that if you keep it at equilibrium, if you don't go all the way down to 0 temperature, there's a learning algorithm, a very simple learning algorithm, which is purely local. You only have to compute the correlations between the pre and post under 2 conditions. 1, when the input's there, and 2, when it's not there, and you subtract the 2, and we demonstrated that that allows you to train multi-layer networks. Not very efficiently, but in principle, it worked. We thought for sure we were going to win the Nobel Prize, that this is how the brain works. And so Jeff gave his prize talk. I was there, and here's what he said. Well, we were wrong about the brain, but it was good enough for a physics prize. Thank you.
Great talk. Please, questions.
Hey, Terry, this is for you or anyone. You said that you needed 100,000 neurons in order to have traveling waves.
Sparse traveling waves.
Sparse traveling waves. So do we see in many, like, smaller animals with smaller brains, like C. elegans or something, like, do we know anything about the comparative, comparative oscillations across species having to do scaling with brain size?
Well, we were focusing on mammals, where they're all sparse. I don't know enough about flies, but you can get a nice traveling wave. Like I say, you just have a chain, and if the synapse is big enough, you'll get it to travel. To get it to be sparse means you have to have many, many more synapses that are small, so that you're not driving every cell every time. And interestingly, you should be able to see the oscillation in the membrane potential. So if you're doing voltage-sensitive dyes, you should be able to see it, even though it's not spiking.
So wait, was I forgot what creature was it, but Barton and Trout even did some modeling. It was traveling waves in olfactory system of, again, I forgot what it was, but it was somewhat on small scale insect-like creature, and it did show some old results about traveling during olfactory coding.
Waves have been seen all over the place. During development, they're very common. And I'm not sure if those are dense or sparse, but, they probably, it'd be interesting to know.
So thanks for a great talk, Jerry. I was going to ask you about the Kuromoto models. So, in 2- and 3-dimensional Kuromoto models, you definitely can have waves, and you can definitely have them with less than 100,000 cells.
Right.
So I'm just wondering, do those waves not meet your criterion for sparsity, or just if you could comment, please, on those models?
So we've actually, Lyle and others now have modeled these traveling waves with Kuromoto models, which are coupled oscillators, basically. And they're models, ecological models for fireflies that synchronize. It turns out if you put time delays between oscillators and connect them up like the cortex is connected with long-range association fibers, you can get beautiful spiral traveling waves. So yeah, you can. There's no doubt that it's part of the nature of neurons. If you inject enough current, they become oscillators, right? They fire at a constant frequency after they adapt a little bit. So in a sense, the Kuromoto model is a good first step. Without the details, all the biophysical details. So yeah, and Lila's gone on to actually do some beautiful analysis, and he shows that you can actually analytically solve nonlinear Kuromoto models, nonlinear coupling, and split it into 2 different equations. You have to go to the complex variables, and there's a linear part that you can analyze in terms of the waves and everything, and then there's the nonlinear part that is changing very slowly in the background. And so, this might be a way to apply it to Hodgkin-Huxley models. We haven't gotten that far yet.
Our entire vocabulary in neuroscience comes from philosophy and psychology. And psychologists define everything with language. And memory is not any different. So when it comes to the first division, you have to come to San Diego and find Larry Squire, who said there's declarative memory, that is something I can declare verbally. That, of course, is not very helpful to define memory for somebody like me who is working with rodents, because they don't communicate verbally. Now, recently we have written a review, an annual review of neuroscience about neurophysiology of memory, and we came up with a different terminology. The first one is very similar to yours. We call it single utility memory, as opposed to multiple utility memory, which is something that is useful for my entire life because I need it all the time, whereas the single utility memory is something that is useful for a while. Now, the interesting thing is there is no time constraint, because single utility memory can last 2 seconds, but it can last for days, because I remember today what is my password at the hotel, but the moment I leave the hotel, it has to be erased. It's no longer utility for my future. So in your STDP model, for me and for many other people who said, oh, this is a kind of Hebbian, I would say Konorskian Hebbian, way how this one passes the other forever. But what is the distinction between long-term memory or multiple utility memory and single utility memory? What happens in the network that said, okay, stop, erase? So this is one question. The second one is, yes, traveling waves are all over. It's easy to demonstrate. putting content into this, you can do it with a model. For me, the most important thing is that every message becomes a message if it is read out. So there has to be an observer, a reader, and so these chaotic patterns and single spiral ways are useful only when a partner structure is coherent with it. What is the solution for you? That how different structures, are they have their own life and they go back and forth, or there are points in time which makes time a neural space? Because time is neural space.
Right, okay, two good questions. Okay, in the first case, there's no reason why with all those synapses, you couldn't have multiple use memories going on at the same time. We know that working memories can hold on to maybe 3 to 4 or 5 different concepts or things that you're working on cognitively. So I think we can accommodate that. But now the decoding problem is more interesting because that in our Trends in Neuroscience paper, we speculate that this is going on in the basal ganglia. We know that the entire cortex projects more or less topographically down to the basal ganglia, the regions in the back of the brain to the back of the basal ganglia and the front of the brain, frontal cortex down to the nucleus accumbens. So there's a rough mapping, but there's now feedback because each one of those areas feeds back to roughly the same area of cortex. But it is distributed over space so that there's a lot of overlap. And what we suggest is that the basal ganglia is the decoder. And in fact, we know it decodes actions because it produces a sequence of actions. Your motor system can only take one action at a time. At some point, you have to reduce it to that. And the basal ganglia, we know, not only does it produce sequences, but actually learns sequences. And with repetition, it becomes better and better, and it does it automatically. You don't need the cortex anymore for the details. So I think that's where I would look for the decoder.
Beautiful talk, thank you, and too many ideas to think about. But there's one aspect of traveling waves that I find it's hard for me to wrap my head around. Maybe you can help, and that is topography. They are going to sweep through a topographical space. So that's fine for vision, for example, but for most of the brain, that's not the case, and for most of knowledge, that's not the case. So do you feel that knowledge is encoded, say, in association areas in a topographical way, or can you think a way of modifying the idea so that the arbitrary associations that characterize your thoughts are encompassed in your model?
So I think we have the answer already for transformers. So transformers, as you know, they're huge, and they have many layers, and you can go in and you can look at them. And now there was a recent analysis, like a 100-page paper that came out of one of the big AI companies. Let's see, what's it called? What? Anthropic, yeah. So Claude. So what they've discovered is if you look at the vectors in different layers, that there's vectors that are pointing in different directions, and that the vectors, if you look at all the words coming in, that the vectors that point in the same direction have similar characteristics, like Golden Gate Bridge. They all point in this direction. And sentiment words like that are good or bad, like lousy, no good, and so forth, they point in another direction. So it looks like there's an organization, like a manifold, that goes through this complex feed-forward network, it's feed-forward, that is clusters. Clusters meaning semantics. It's semantically organized. And we actually see that in the cortex. Jack Gallant has shown that, he's language, and he shows that, if you use fMRI, you can sort of see regions that are kind of like that. I mean, it's very low res, but, I don't see why that's a difficult problem. In other words, we know that for any given action that you take, you're actually only activating a small subset of all the neurons. It's a low-dimensional subspace, low rank. And you can fit many of those low-rank activity patterns in the very same large-scale network if you actually are very efficient about being able to put them together without interfering with each other. Maybe, we're actually, I think we're over time. One last question?
Yeah, mine was if I like that you put on your talk like all these predictions. I was wondering, do you have specific experiments in mind that people could do like for specific tasks and like tests like these spatial-temporal coding that we could like make progress in trying to test these?
Yeah, I have a whole list of them. It's actually more difficult to study this in animals because you can't talk to them than in humans. So I should talk to Eric about this because Eric has access to these patients. So there's all kinds of working memory tasks. And if we have enough electrodes in different places, we should be able to see traveling waves and try to see whether they're during different aspects of the task, like end back. You give a bunch of inputs and you have to say what was the third back. Right, that's a good example of a working memory task. But there's a dozen tasks like that. So yeah, all of that can be tested in humans.
Okay, well, thanks.